Morning, depending on where you're joining from, and welcome to the AgriFutures Chicken Meat Program webinar series. AgriFutures acknowledges the First Nations people of Australia as the traditional custodians on the lands, waters on which we live, learn and work. We pay our respects to past, present and future elders on these nations and any First Nations people on the webinar today. In particular, I'd like to acknowledge the Ngunnawal people of Australia, the traditional custodians of the lands and waters where I am hosting this webinar from today. I would equally like to extend this acknowledgement to the audience on behalf of our speakers presenting today and the respective countries that they are presenting from. So welcome once again, and it's great to have your company. Before we get into exploring some of the research being featured in the webinar, a few housekeeping items. We will be recording today's session, and that recording has started and making that available through online channels. As I run through the housekeeping items, there will be an introductory poll that pops up on the screen with some questions, and that's just for us to gauge who's in this virtual room, and we'd be grateful for your responses. As this is a webinar, you may have noticed your video and audio functionality are not enabled, and the reason for this is that we can best utilise that Zoom screen real estate to view the shared content from our presenters, and it also allows us to minimise any background noise and distractions to our speakers. We will have time for questions and answers at the conclusion of the webinar content, and then in the meantime there is the dedicated Q&A chat function to ask any questions over the course of the webinar as they pop up, and that's located, I think, along the bottom of your Zoom screens. So quick introduction to myself. My name is Sarika Pandya. I am the manager, part of the broader AgriFutures Levied and Emerging Industries team, and I specifically and solely look after the Chicken Meat Research, Development and Extension Program. Now, this webinar, I think, is the 8th in the series, and we host expert presenters to provide you with the latest information on topics and research that's relevant to industry, as well as the industry's research priorities. Now, today's content features the project titled Sustainable Precision Feeding in Broiler Chickens in Australia, and I will not use that title again, but it is more commonly known as the Consortium, which has brought together poultry industry partners and research providers to investigate how chicken meat producers can use nutrition to optimise gut health, welfare and sustainability outcomes. Now, this consortium is jointly funded by AgriFutures Australia and a range of other organisations, making the consortium big in scale and potential impact. It comprises of 6 sub-programs, and we will hear from the leads of sub-programs 1 and 2 during today's webinar. Now, the consortium has successfully completed its first 2 years of the research programs, and this achievement marks the halfway point of the consortium. And again, its aim is to advance the poultry industry through innovative research and development. So I hope you enjoy today's content. Now, I'll start by introducing our first speaker, Eugeni, and whilst I'm doing that, I'll invite you to turn on your video and begin sharing your slides. Professor Eugeni Roura is a nutritionist with interests in digestive physiology and gut health, chemosensory science and transgenerational mechanisms relevant to chickens, pigs and humans. Having developed a 16-year career in the feed and food industries, he joined the University of Queensland, where he currently leads the Nutrition and Chemosensory Science Group, part of the Queensland Alliance for Agriculture and Food Innovation. QAFI's main research areas are in animal science and include appetite modulation in chickens and pigs, nutritional interventions to boost embryonic or fetal development, including in ovo, and strategies to improve sustainability of chicken meat, egg and pork production. Eugeni is the director of the Chicken Meat Consortium and also the lead for sub-program 1, and will be informing us today on maternal programming and amino acid precision feeding in broiler chickens. Over to you, Eugeni.
Thanks so much, Sarika, and welcome everyone to this update of as Sarika mentioned, the Chicken Meat Consortium for Nutrition, Gut Health and Environment. If someone can confirm that everything's fine and that you can see my slide, that would be great. I'm assuming it is. So first, an acknowledgement to, as Sarika already mentioned, thanks for the introduction, Sarika, of this consortium, which is really a master piece of collaboration, if you want, in the sense that involves under the initiative of the AgriFutures chicken meat program. It involves 5 research providers. In addition to the University of Queensland, we have as main partners the University of Sydney, Massey University, Central Queensland University, and the Queensland DPI, Department of Primary Industries. We also have as principal partners several industry stakeholders, including Aviagen, DSM, Firmenich, Eurolysine, Lacta, Lallman, the Poultry Research Foundation, and the World Poultry Science Association of New Zealand. To all of them, really my gratitude, our gratitude, because without them, obviously we wouldn't be here. So thank you so much for all this group that it's delivering, I believe, a fantastic outcome. And I'm going to try to summarize a little bit where we are, at least in Program 1. But let me first remind everyone of what this whole consortium is about. And very briefly, what the consortium is about is initially understanding that there are limitations that could be improved on matters such as the sustainability. We do all keep in mind that we're still using a lot of soybean meal in our diets, and soybean meal is important. In some cases, this soybean meal import has been related to deforestation. There's obviously a significant carbon footprint because of the importation. So that's one of the important points of the consortium. Can we dilute that reliability on soybean meal? Now, what goes in the chicken, then it needs to be substituted potentially by some of the local ingredients that we can find in Australia and New Zealand. So from canola to some of the grains we know, and even some other alternative protein sources, non-vegetable. This basically, this what goes in this nutrition part of the program, which is including 3 of the 6 programs of the consortium. Program 1, which is the one that I'm leading, will be looking at how can we intervene in broiler breeders and how can we intervene during embryonic development to make these things happen. In a long story short, then we have program 2, which is looking at the digestibility of some of these ingredients, and I will let Professor Mike Gidley to further explain on what they are on this. Program 3 is led by the University of Sydney and is all about alternative feed ingredients, feed formulations. Indeed, together with Massey University, they are collaborating in the same aspect. Then we have Program 4, which is led by Dana Stanley at Central Queensland University. I forgot to mention that Program 3 is led by Sonia Liu at the University of Sydney and Tim Wester from Massey University. Program 4 is Dana Stanley, Central Queensland University, and it's about gut health. It's about seeding that early gut in the chicken to make sure that gut health evolves since day 1. Program 5 is led by the Department of Primary Industries of Queensland, former DAF, and the program leader is Dr. Mark Dunlop. And it's all about how do we improve the in-shed environment. And very important to the philosophy, if you want, of the consortium is Program 6. And as you see, Program 6 goes all around the other 5 programs. And that's what it exactly is an integration program of the overall consortium. Now, I'm going to focus in Program 1, which is the program that I'm leading. And I will first acknowledge the contribution of a number of scientists that are helping us with several of the sub-programs that we have in Program 1. I would like to acknowledge Dr. Arun Kumar, Anna Nguyen, Lida Omaleki, Connie Turney, and Drs. Eli Asatsume, and Marta Navarro. So to all of them, thank you for all their contributions. That's what it is about. If you have a look at the cartoon that I have put in here, it's about that transition. It's about this understanding the impact of breeder nutrition on embryonic development and then the early seeding, etc. And all about getting a robust day one post-hatch chick ready to go. Let me go into a little bit more of detail. What are the challenges? I think it will be no surprise to anyone in this group that probably one of the main challenges in broiler production is getting that day one chick with a robust, healthy gut, immune system, etc. And that's really what program one is about, if you allow me to say it in one sentence, is understanding and trying to improve the production of post-hatch very young chicks. That's what we're trying to do. And how do we do that? Well, we're trying to do that through dietary interventions in breeders, but also in ovo manipulations, as well as early feeding strategies. I'll get a little bit more into, briefly, I'll review the 4 sub-programs that are involved in this program one. Credit to PhD candidate Bipul Chandra Ray. Bipul is in fact with Dr. Arun Kumar leading this broiler breeder program. Now, the broiler breeder program has a slightly different spin than most of the broiler breeder research, research projects that I've been aware of. And the spin, if you want, is because we're looking at the nutrition of the broiler breeder in terms of how do we nourish the embryo. So we probably at this point, nutritionists have done a very good job in having very good nutritional programs for broiler breeders. But is that broiler breeder nutrition having the impact that we expect on the development of the fertile egg? And you may agree with me, especially when I'll show you some data, that this may not be the case. So we're feeding broiler breeders for the broiler breeder production, but basically we're not always thinking of how do we nourish the developing embryo. So this first experiment that I'm showing you today is about an experiment that we did a few months ago. It's a factorial design, as you see, where we looked at 3 different crude protein levels in broiler breeder diets. And those 3 protein levels were aiming at somehow a standard level, 14%, but also we went a little bit higher and a little bit lower than what normal practices would recommend. Now, these 3 different levels of broiler breeders were then split into each one of those 3 different levels in the progeny. And those 3 different levels equally would be somehow around what's a standard, a little bit higher and a little bit lower. And that's across the 3 treatments of each broiler breeder. Therefore, that's the 3x3 factorial design. So we have 9 treatments indeed. Now, I'm going to focus because in interest of time, I wouldn't be able to review all the results we have, but certainly I want to make a message, which is I'm going to look at what is the impact of these low crude protein diets basically in broiler breeders, how does that impact the performance of the breeder, but also the development of the embryo. And the bottom line of the data that I have chosen to show is that when we looked at breeder performance in general, fertility, production, laying rates, etc., we could easily go to low crude protein diets as low as 12% and have practically 0 impact or very little impact. And that's what I'm trying to show with the fertility rate, etc. Now, hatchability, when it comes to embryonic development, it was not significantly affected, but you already see here that the 12% crude protein level, which is the lowest crude protein level, had a bit of an impact, not significantly different on hatchability rates. Now, when we actually look at the embryos, that's when the big differences arise. And you can see that we had a significant increase in deaths in shell. We have a significant decrease in the intestinal weights of day-old chicks born from breeders fed the lowest crude protein diet, 12%. And as you see, these differences were highly significant with a p-value of less than 0.01. Now, this is important, and I wanted to really leave that message today, that when we're feeding broiler breeders, we might be having a very balanced diet for the broiler breeder, but this, in our view, may not guarantee always that the embryo is getting all the nourishment that it's needed. And that's one of the take-home message of today. I apologize for such a busy slide. I realized that it is too busy, but I'm gonna summarize it. I'm gonna just try to make it simple. What I'm actually trying to show you is why did we see a negative impact on the intestinal development in chickens which moms were fed a 12% crude protein diet. And as you see, the reason why this reduced intestinal weight was observed is basically because a number of metabolic pathways, and I'm not going to get into the detail, otherwise we would be here until tomorrow, but please take the summary that some of the main fatty acid pathways were disrupted. So we did see fatty acid degradation. We did see exceptionally high oxidative stress going on in these young chicks, day one, that actually had an underdeveloped intestinal tract, right? Now, how can we overcome this? And this links, therefore, with other parts of our research programs. And I'm jumping to a program that is actually, it's a program 1.4, which links the broiler breeder diet with innate preferences in day one chicks. And I would really like to call your attention on this data because, in my opinion, what we have observed, the way we can manipulate innate preferences in chickens is absolutely incredible. Now, see what we did. This is actually a second experiment of a series. I'm not even going to show the experiment number one, which had a very similar result. But what we're actually trying to do is we're trying to control the broiler breeder diet, which is not too far from a standard diet, but simplified maybe. It's a wheat, soybean, canola oil. And then we would take the progeny of these broiler breeders and offer them either a wheat-based diet very similar to the diet that the breeders were eating, so again, a wheat soybean diet, or offer them a corn-based diet, something that was completely different to what what mom was eating, basically. And the corn-based diet was a corn, canola meal, peas, lupins, etc. Now, these diets were isocaloric, they were isoaminoacidic in terms of essential amino acids, so they were really balanced. The only difference was the feed formulation itself, the ingredients. Now, have a look at the results. When we give the choice of the day one chicken what diet they prefer, we gave them a choice between wheat and wheat. So this is no choice, basically. And effectively, that proved the concept that the double choice was working. There was no preference because they were choosing between exactly the same diet. So wheat versus wheat, no preference. Corn versus corn, the same, no preference, as you would expect. But when we offered them the choice between a wheat and a corn-based diet, so remember that the wheat was the diet that mom was eating, right? The results were astonishing. Look at these 4 asterisks, meaning that the p-value is less than 0.0001. Very statistically significant. That's the accumulated of 7 days, so the first week post-hatch. If we look at the evolution on a daily basis, you will see that even on day one, but certainly significant on day 2 at p less than 0.01, you would see that there is a clear preference of the chickens for the wheat diet compared to the corn diet. Now, equally relevant, in my opinion, is the fact that those chickens offered the wheat double choice, which is a controlled diet, were actually growing significantly more because they were eating much more than the corn, while the wheat corn, the double choice, truly, they were somewhere in between. Now, why is that happening? That's happening probably because of the transmission, what we call this intergenerational conditioning of what mom is eating. Mom is eating a diet. We have measured the plant volatiles that are present in the diets of the breeder. And we see how these volatile compounds are transferred to the plasma of the laying hen. And from the laying hen, we can find the traces of those volatile compounds into the developing embryo. So we believe that that's the way how mom is communicating with the developing embryo 20 days later, that actually sends the message that that's what you should be eating, that's safe food. So day one chick, they hatch, and the first thing they look for is that this familiarity of volatile compounds that were found basically in mom's diet. Now, another thing that we believe it's important when it comes to preferences in double choice, and we're still talking about double choice in very early life in chickens, is the actually relevant question of what essential amino acids might be most limiting. And we've run, as part of this program 1.4, not only we've studied volatile compounds, we have also studied 20 amino acids, as you can see here. You see them, the 10 essential amino acids. And as you would expect, the diets were formulated so that we were giving this the choice between a diet with an excess of each one of the amino acid and a choice with a diet that was deficient, 20% deficient. The theory is, because they were formulated based on the ideal protein ratio, the idea is that the chicken would be eating equally from both, so that the 20% excess would balance the 20% deficiency. And that was the case for all of the amino acids, except for arginine. So there's something in this experiment, and these diets were low protein diets, that tells us that arginine is possibly a limiting amino acid, especially early in the life of the chicken, which links really nicely with what Nadira has been looking at, PhD candidate Nadira Disanayaka. We looked at supplementing the embryo Inovo with amino acids, and we've tested arginine. I'll tell you why. And we've tested now some other amino acids. Now, arginine, when we look at the ideal protein ratio, or the ratio that you will find in starter diets, every amino acid in the egg is at least at the level required, except arginine. Arginine is the only essential amino acid in the egg that it's below what you would think is the ideal protein ratio. So it makes a lot of sense that we're actually selecting a supplementation of arginine in ovo, and that's what we did. So we had a dose response, including 0 all the way up to 100 milligrams per egg. And the number of eggs was 50 per treatment, and we injected that in the amniotic fluid, as it is the standard for Inovo injections. Day is the 17.5, because that's also the day then that Marek's disease vaccine is injected, and working at that 17.5 day of embryo development, hopefully would allow adoption by industry being a little bit easier. Now let's see the results of the supplementation of the amino acids, the amino acid of arginine. And actually, I'm not going to show you performance again in interest of time, but it was very clear that above 60 milligrams per egg, arginine was toxic. We had a very negative impact on hatchability and on embryonic development when we were injecting more than 60 grams. When we were injected below 60 grams, we didn't have any significant difference. So we looked at what was happening with these injections. In fact, we chose the 80 milligram egg, so just a little bit higher than the minimal toxic response. And we did see, among other gene expression differences. One of the main was related to nutrient transport. So somehow the excess of arginine had an impact on a number of pathways that ultimately had a negative impact on growth and performance of the chick. So going on the amino acid side of things, we didn't have much of a good results in injecting amino acids in ovo, but we're hoping that some of these amino acids may actually help the day one chick develop a healthy microbiota. And why do we think that? Well, this is subprogram 1.3, which is managed by Lidao Maleki and Connie Turney. Thank you both for your excellent work. And the PhD candidate is David Suiki Chow. This is a phenomenal program. The results we're obtaining are out of this world, fantastic. And what it is about is exploring, if you want, potential amino acid requirements in the microbiota. So do all the bacteria have the same needs for dietary amino acids? And the answer is no. Some of these bacteria might be able to synthesize almost any amino acid, so they probably don't have amino acid requirements. But some others actually do. They have amino acid requirements that they need to get from the environment. So David is doing this systematic study on looking at the impact of different amino acids on the growth of the bacteria. He has now finished all the pathogenic bacteria in the list you have here, and he's starting to look at beneficial bacteria. So the objective, of course, is can we supplement amino acids early in the life of the chicken to develop a balanced microflora? Just a hint of what we're finding. And this is in vitro cultures. And as you see, we looked at the 20 proteinogenic amino acids, and we see the impact of these amino acids on the growth curve of each one of the pathogenic bacteria. I've chosen to show you E. coli. All the work, of course, is done by David, so I'm borrowing his marvelous stuff. Thank you, David. And as you've seen here, cysteine and histidine had a bit of a negative impact. Actually, cysteine, a very important impact inhibiting growth. Histidine, a little bit of a delay in the start of the slope of the growing curve, while most of the other amino acids had no impact. I'm going to show you a table from David which summarizes what amino acids are giving consistent results in terms of either inhibiting or promoting the growth of these pathogenic bacteria. And we're talking about Clostridium perfringens, coli, Enterococcus, Staphylococcus aureus, and Enterica. So as you see in this really well-placed table, main impact would be from cysteine, which is an inhibitor of pathogenic bacteria growth, while glutamine, glutamic acid, proline are the main ones promoting effects. Then we have other single aspects that we could go into the detail. I'm not going to go through that. But this is important data because I believe it sets the scene for what's coming very soon in that role of we call aminobiotics. So it's like a prebiotic role of amino acids, right? Now, a couple of slides to finish my presentation. And this is to introduce you a couple of the most novel aspects of our research in Program 1. Francisca, a PhD candidate, she's looking at how can we improve the development of the gut. And more precisely, we're looking at mechanisms that relate to cell differentiation, how the stem cells here in the crypt of the villi how do they choose to become either an enterocyte or an endocrine cell or a goblet cell, etc., etc. And we want to be able, at the end of the day, to modulate that maturation, if you want, and migration of these cells, and being able to improve or increase, if possible, either goblet cells or enterocytes as it is advantageous at any given time. But we do know, and we have proof of concepts, that through the diet, we can change that maturation, and that's exactly what we're trying to do. This photo to the right is a beautiful photograph that shows all the goblet cells present in the ileum of the chicken, which is a brilliant illustration of what Francisca is doing. And future work, I'm going to finish with this. Future work, we're going to try to get a bit more of the insight into how broiler breeder nutrition can have an impact on muscle cell development, muscle fibers in the embryo. And that's something that we are trying to start with a new master's student, Benny, where we will be looking together with program 1.1, so BPOOL. We're going to be looking at the impact of broiler breeder nutrition on the development of muscle fibers and potentially some muscle myopathies, or how to prevent them. And with that, I would like to thank everyone listening. Thank you so much for attending this webinar. Happy to take questions. I think we're going to do that at the end. And also thanking all the team from the Bioscience Precinct, Connie and LIDA team, our team, the Nutrition and Chemosensory Science Group. Thank you, everyone, for your attention.
Thank you very much, Eugenie, and a lot of content from sort of the 4 sub programs within program one to cover there. As you'd sort of alluded to at the end, we'll keep some time at the end for questions. So if there are any that you do think of, even during Mike's presentation, feel free to put them into the Q&A function. Eugenie, I'll put you on notice. There is one comment in the chat. I'll let you have a look and ponder on that, and I might come back to that at the end and see if you've got any a comment in response to that. But with that, I'm going to move over to introducing our second speaker for today's webinar. So Mike, I'll invite you to start sharing your screen. Professor Mike Gidley is a food scientist by background and looks to use insights from human foods and nutrition to understand and optimize animal feeds and nutrition. He's interested in plant tissues, including grains and legumes, and their polysaccharide assemblies such as plant cell walls and starch granules, and particularly the way that these structures are assembled in nature, then sort of disassembled during feed manufacturing, and then later in digestion or during digestion. His field of research involves the use of spectroscopic, microscopic, and material analysis of natural materials and model systems to provide targets for raw materials and processes which enhance feed and nutritional properties. And during this next segment of the webinar, we'll hear from Mike about carbohydrate and amino acid precision feeding. Over to you, Mike.
The time, if you let me, Sarika, to have a question as you get started, Mike. So the question is about whether we should be having another control diet in broiler breeders to be able to contrast our findings. And basically, I agree, this is a very good observation. And yes, we are working on the next experiment to have actually the 2 diets, and that will be a complete factorial design. We didn't have access to a factorial design at that time. Mike, you ready to go?
Let me see. How are we doing now?
I think better. Yes.
Got you, Mike. Perfect.
Okay. All right. Thank you. Thank you for filling in, Eugenie, as well. So, hello, everybody. So, program 2, following on from what Eugenie was saying, I think in a nutshell, what our role here is to say that there's a lot that's understood about the molecular compositions that you would like to see in a diet for chicken meat. And Eugenie's alluded to that in terms of amino acid ratios and so on. But there's probably less understood about the form in which we give these nutrients to the birds. And so that's really the main topic we're looking at, is how do we control the way in which we provide the carbohydrates and proteins to birds and the impact that has on their nutritional outcomes. So as Eugenie has said, we have a whole series of programs in the consortium. and following on from what he described as the intergenerational strategy, we're looking really through the growth of the chickens themselves. So we're not looking at the broiler breeders or in ovo. We're looking at the grow out process. And our main aim is to try and understand how do we best put together diet ingredients that provide the two main energy sources, carbohydrate and protein, so that we get rapid growth, but we also partition enough of the non-digestible carbohydrate that we can nourish a healthy gut microbiota, limiting pathogen infection, limiting the needs for interventions, and so on. So just a little bit of orientation, if you like. So in terms of the way in which birds handle the feed that they are fed, we've really got three stages. So we've got what I call a preparation for digestion, which is a hydration process, a grinding in the proventriculus and gizzard, addition of some enzymes, some pH control, and so on. And this is a particular feature of the birds, that the feed that they eat is significantly ground in their upper digestive tract before it gets into the main enzymic digestion processes in the small intestine, where the pancreatic secretions of amylase, protease, and lipase break down the three main energy sources of starch, protein, and triglycerides, and where the enzyme hydrolysis products, other minor components, and water are absorbed. And then if any of the energy or other components survive that process, then by the time you get to the openings to the ceca, then there's the opportunity for that residual material to enter the ceca and alter the microbiota profile, which can have a major impact not only on the bird itself, but also on the carryover of any pathogens into the foods, as well as being a harvesting of additional water. So some of the issues we're talking about here. So in the first stage, it's really about softening of the feed particles and some size reduction that facilitates enzyme access and is the single factor which controls the rate of the subsequent enzyme digestion. In the small intestine, digestion rate versus passage rate. So there's a certain time that is available for digestion of the feed as it passes through the bird, and if digestion occurs within that time frame, then it happens. If it's too slow for that time frame, then the bird passes residual feed further down towards the ceca and the colon. So the digestion rate versus passage rate really controls the dynamics of how energy, protein, and lipid are absorbed, as well as the nature of the undigested material that's potentially available for as a feedstock for nourishing the cecal microbiota. And then finally, depending on the time frame of fermentation versus excretion events, then the amount of energy that's recovered from microbial fermentation in the cecum, the amount of water absorption that occurs compared to what is excreted is controlled as well. So the dynamics are really important here. So composition, of course, is important in terms of feeds, but understanding the dynamics in how those components are utilized by the bird is the focus of our work. So what are we doing? Our approach, I'll talk to you about ex vivo analysis, so looking at digester from different parts of the digestive tract to understand the consequences of post-gizzard particle sizes, and to get some insight on what the nature of the digestion-resistant feed is at the ileum and colon. And then to use that information to refine in vitro digestion processes which are well established in humans as good indicators of endpoints and rates of digestion, and using that as a way to screen as large a number of variables as we wish, but particularly looking at the physical form of the feed. So that's as influenced by particle size distribution, heat treatment during processing, and the addition of exogenous enzymes. And then taking the consequences of in vitro digestion and saying, if that digestion-resistant feed is available, then how does that impact particularly the cecal microflora and the opportunities for enhanced gut health and pathogen resilience? And this can all link through, we will link this through later in the year to an in vivo trial where we'll look at test some specific hypotheses that arise out of the in vitro work, and we'll apply the principles we learned in the integration effort that Eugeni emphasized as a key part of the consortium. We want to bring all of our different tools to bear to tackle this single issue. So the two areas we'll talk about, one is about optimizing, how to set about optimizing in vitro digestion of carbohydrate and protein sources, and the second is for enhanced microbiota pathogen tolerance from in vitro fermentation. So in looking at how do we develop an in vitro digestion process, it's really about matching ex vivo samples. So we want to do a laboratory test at the bottom left that gets to the same endpoint as happens in the bird. We do not want, we do not want to make a chicken digestive tract in the lab. We want to get to the same endpoint. That's the sole purpose of the in vitro method. So we're not trying to represent the biology, we're trying to represent the biochemistry and the biophysics. So what we did was collect samples from trials that were going on anyway, from birds, particularly of different ages, 7, 28, and 42 days, different points in the digestive tract, and then analyze them microscopically to just see what they look like, particularly quantifying their particle size distributions and their moisture contents, and using particularly total starch content, because all the starch that you find in the digestive tract has come from the feed. If you measure protein content, the allowance for endogenous protein makes it somewhat more difficult, but starch is only coming from the feed, so it's a good marker for what's happening in terms of feed digestibility. And so we can use our information on particle size and moisture content to set the model parameters, making sure we're dealing with similar particle sizes of the feeds that we're going to test and at the appropriate moisture content, and then using the starch and protein residuals as the model results that we want to get to the same endpoint from in vitro. So let's look at this particle size. The way we do it is, I've done it here, is by just using a stack of sieves. So we're looking at particles, we're using, for instance, mash feeds here. The largest size fraction is bigger than 1.18 millimeters. That's the light blue at the bottom of the bars, up to the smallest one, which is not really present in the mash feed, which is less than 53 microns. So you'll see that there's a distribution in the mash feed. Then it's mostly greater than 1 millimeter and between 0.5 and 1 millimeter. In the gizzard, because this is actually in the gizzard rather than post-gizzard, there's actually more larger particles because they're the ones that stay longest and which have require more processing to pass through. When you get through to the jejunum, then you can see that the large particles are reduced significantly from the gizzard to the jejunum, which is exactly what you would expect. But still, there are a significant fraction of particles that are certainly more than 400 microns. So about half of the particles are bigger than 400 microns, which is a pretty large size for a chicken, and only a small amount is less than 200 microns. What's interesting is that doesn't change greatly when you go from the middle, the jejunum, towards the end of the small intestine. The ileal particle size distribution is approximately similar. If anything, there's a little bit more larger particles because they're the ones which are slower digested, and so they're a bit more concentrated at the ileum. But the fact that we're getting really rather similar patterns here from 7, 28 and 42-day-old birds is reassuring. It says that at least over that time frame, we can have some confidence that a single in vitro method is pretty good at making predictions about relative digestibilities, irrespective of age, from 7 to 42 days. In terms of dry matter, so in the jejunum and ileum, it's pretty consistent around 20%. So which is good because that's pretty much what is used in the human in vitro digestion method that's well established. So we will match that. And then if we measure, in this case, starch content, you can see that there is that goes down, of course, and then you can use the residual starch at the ileum as a really good marker of the digestion resistant material ex vivo, and that's what we will be matching in vitro. So the summary of that is we think we can use a single age-independent in vitro model, and knowing those particle size distributions, we've found that just using a simple coffee grinder under certain conditions, we can get pretty far in terms of replicating the particle size distribution. So starting with the feed and putting it through a coffee grinder in a certain way, ends up with a particle size distribution that's rather similar to what you see at the jejunum. So that's also a very good practical adjunct that we can use, because this is not what you do in the human case, because in humans, the chewing you can easily mimic. We don't have a gizzard, clearly. So once we've done this further validated, then we can run through the lots of combinations of feed ingredients, their particle sizes, and exogenous enzymes to understand how to what tools we've got really to improve digestibility independent of the chemical composition. And as I've mentioned before, that digestion model can then also be used to simulate the digester that exit the small intestine and are available for some fermentation in the ceca. Which takes me on to the second part. So number 2 is about understanding how we can learn something about the way in which the cecal microbiota responds to the undigested feed that it has the opportunity to sample. So the way we do this is, again, using a model that's well established in both humans and pigs of in vitro fermentation. Our standard first pass analysis is just measuring the gas that's evolved. So we have this setup that you can see on the right, where we have a large number of bottles, so we can do the biological replicates and run many different samples all at once. What we do then is we take a sample, in this case of cecal microbiota ex vivo, diluted in saline, use a basal medium where the main carbon source is the substrate that we want to test. So we're saying, okay, this is what would happen if the substrate we want to test is what the microbes have to grow on. So that is what we're testing in our in vitro fermentation measures. So in a first experiment, we used in vitro digested wheat bran, and some standards, the wheat bran without digestion, wheat starch, maize starch, and some standard fibers, just to get an idea of the range of responses that we might get. So we took cecal microbiota from both 7-day and 42-day-old birds from different experiments. So it's not a single experiment, but examples of microbiota from 2 sources. And found, I guess the first interesting thing is the fact that all of these lines go up means that there is fermentation of all substrates by both sets of microbiota. So there are none of these sort of standard feed components are resistant to fermentation, but there's a clear difference in the relative fermentability. For the 2 samples we had, one happened to be from 7-day-old chicks, one happened to be 42-day-old chicks. But the most important thing is that they responded to every single one of these potential undigested feed substrates. Interestingly, they all produced one of the main markers for gut immune health, which is the short-chain fatty acids, and really pretty much the same, irrespective of whether we had a 7-day-old or 42-day-old. If we took the total short-chain fatty acids, but they're different patterns of the 3 main types, acetate, propionate, and butyrate. So that the 42-day-old had produced a lot more propionate, which is what you'd expect, because that's a marker for an animal that's adapted to a high fiber type diet, which they will have had because of the undigested fibers. Whereas acetate is a marker for less adapted fiber, more starch and protein fermentation, which is what you see more for the 7-day-olds. What that also is reflected in is, again, please don't look at the details here, but just to say, on the left-hand side with the purple column, that's the 7-day-old. On the right-hand side, the green column, that is the 42-day-old. And these are just the genuses which are present in these microorganisms, in these microbiota. And at the bottom, you'll see one of the lines is inocular. That's what we started with, and all the other ones are what happened after they've been fermented with the individual substrates. So clear difference between the different ages, and that's not surprising. And some small variations even after 48 hours of in vitro fermentation, showing that we can shift the microbiota a certain amount, although mostly that microbiota reshaping will take over more than 1 or 2 days. Maybe 7 or 10 days is a more realistic time frame to really reshape the microbiota. Where we're going next with this is to go deeper in that microbiome analysis to identify endogenous pathogens, particularly E. cecorum, Faecalis, and E. coli. Our colleagues here at UQ have previously found these organisms in previous deep analyses of chicken cecal microbiome. So we're confident of finding them, and then you can use those. You can then quantify these organisms using quantitative PCR and get an idea then of how well individual undigested feeds are able to either nourish or not nourish these harmful pathogens that we want to try and control. However, the other thing that we really need to consider is back to the particle sizes, because it's becoming clear that there is a pretty strict particle size filter on cecal fermentation. I'll show you what I mean by that. We'd noticed that by looking at the cecal contents, very sort of pasty, very little evidence for any large particles in there, which completely contrasts with what was present in the small intestine and indeed in the colon. And a paper recently came out just a few weeks ago that was also discussed at the APSS in Sydney recently, that entirely consistent with what we find as well. So what this is showing is a different way of showing the particle sizes. It's by a light scattering method rather than a sieving method. But that sort of long, low hump is the feeds that are present in at the ileum, starter, grower, and finisher. And you can see it's the same sort of distribution. We were getting hundreds of microns up to something over 1 millimeter. But in green to the left and amplified in the box in the center, it's a particle size distribution that was found in the ceca at the same time as the ileal distributions that are shown in the big graph. And you can see there's nothing below, very little below 150, and actually not much above 100 microns. And at least for the early ceca, most of them less than 20 microns. So this is quite an eye-opener for us, because I think this tells us quite a lot about where fermentation may or may not occur. Because if we take even that sort of fairly generous particle size cutoff of 200 microns, that little bar on the right is showing you those particle sizes that are less than 200 microns. At the ileum, and consistently we find between 15 and 20% of particles at the ileum are smaller. So the inference from that is that 80 to 85% of what's at the ileum does not go into the ceca. And this is similar estimates were reported by that paper I just showed. So this is causing us to think about sort of whether there's only a very selective part of the feed which ever gets fermented by the cecal microbiota. It also begs questions about the roles of microbiota in other parts of the digestive tract. Although the densities of microbiota are much less. In the ceca, it's a super microbiota. Elsewhere, it's a thin film. And so we're not sure yet what this means, but the implication is that it's only a small fraction of the undigested feed which is ever available to the cecal fermentation. So that's it from me. So we'll continue to work on optimizing in vitro digestion so that we can give ideas about how to use feed technology primarily. So control of particle size and enzyme addition as an additional tool as well as the chemical composition of feeds to influence the nourishing of the birds. And how we can use the ability of undigested feed residues to promote the beneficial bacteria and limit potentially pathogenic strains from developing. So as I said before, this is all part of an integrated program. So we have a great opportunity to try and get learnings across the whole range of the growth cycle in chicken meat production. That's it from me. Thank you so much. Very happy to answer any questions.
Fantastic. Thank you very much, Mike. I'll ask you to keep your video on and Eugenie to turn your video on. We don't have many. We only have a few minutes before we sort of hit the hour mark. However, I will ask a couple of questions. One which was received prior to the webinar, sort of during that registration process from an attendee, and I'll sort of direct this both to you, Eugenie and Mike. And the query from this individual was, what are the best meal substitutes to provide indigestible fibre for layer chickens in their diet? And maybe the follow-on question is this known?
Short answer, no, it's not known directly. You inevitably get indigestible fibre every time you feed grains. So it's really a question of what grains have you got in the feed, because that is the main source that is an inevitable source of indigestible fibre. Whether you choose to add extra is another matter. But first of all, you'd have to look at the choice of grains, because that influences the nature of the fibre that is produced. I'm not aware of any systematic study of that, though.
Wonderful. No, thank you very much for the response, and hopefully the individual that's asked it is either here with us today or is able to view the recording to hear the response. And I'll use this opportunity to ask the one question that has come through the Q&A chat function. I'll direct this again to both of you, and it's about digestion timelines and specifically how long food remains in the different stages of digestion, from ingestion to excretion. So specifically the approximate time the food spends in each of the major sites of digestion. I'd imagine diet, age, and bird health might be likely factors that have a role to play, but I'll hand over to you, the experts, to respond to that query.
I mean, my one answer is that there's not a single number, that there's a very wide range here. So we saw in our data for the particle sizes in the gizzard that the smaller particles probably go through the digestive process more rapidly than the larger particles, because they get retained in the gizzard for further grinding. So a single number is misleading. And I think it's, I'm going to go back to some human an analog here, that one of the main ways in which we try and reformulate foods so that we keep people fuller for longer is to have that drawn out digestion process. So I think that there is something here also for chickens, that if we want to manage the time frames over which energy is absorbed and muscle is developed, we have the opportunity to think about doing that. But so having it too fast is as much a problem as having it too slow, I guess is what I'm trying to say, because you want to have the other metabolic processes you're interested in stimulating. They need to be in synchrony in terms of time frame with the availability of energy and the building blocks, the amino acids. Yeah, that's probably what I would say.
If I may add one comment, Sarika, I know we're running out of time, but you might mentioned all these new findings about the particle size getting into the Zika, and that does change a bit the scenario, in my view, right? So it's interesting to see that particle size above 200 millimeters would not
Micrometers
would not get in the Zika, right? That really changes the flow of the content.
Thank you very much, both. If there are any other questions, noting that we've just gone over 3 p.m., I will invite any other questions to still be sent through the chat or to the chicken meat program email, the one that you've been receiving all of the invites from, and where possible we'll get these questions to our experts here today and provide responses to them. So what I'll do now is I think I'll just quickly run into the thank you and the wrap up for the webinar. So I'd like to bring your attention to a post-webinar survey that will show up on the screen sort of very shortly. And whilst that's there, and whilst people are responding to that, I'd like to thank a really big thank you to both of our presenters, so Eugeni and Mike, for your time today, for their time in preparing your presentations. Taking the time to share this research that you have underway is really, really important for the industry, and we've had a lot of interest in this particular webinar content. And as the consortium enters its second half, we're looking forward to hearing more about how it will continue to deliver those tangible benefits to the poultry industry. And today we heard from 2 of the sub-programs, and at the start I mentioned that there were 6, so don't worry, the other 4 sub-programs will be featured part of the webinar series, and we'll look to get the programs back as well as there is more to share. So stay tuned for more updates. And of course, I'd like to thank all of our audience today, those who have registered and were able to join, but even those that have registered but will view the recording sort of later on in their own time. So thank you very much for your time, for your responses to the survey. I'd like to wish you a fantastic rest of your Tuesday, and we shall see you next time. Bye for now.